Hi, hello, welcome to my channel, Dr. Srivani Sesham, presenting to you another important topic, giant chromosomes, the Lamprecht and Polytene chromosomes. So, if you have not yet subscribed to my channel, please do like, share, and subscribe to my channel. Let us now get into the topic giant chromosomes. What are giant chromosomes? The giant chromosomes are the chromosomes which are very large in size as compared to normal chromosomes. They are also called as mega chromosomes. These chromosomes are very long and thick in appearance during metaphase than normal chromosomes. Hence, they are called as giant chromosomes. The giant chromosomes are the Polytene and Lamprecht chromosomes. Lamprecht chromosomes are a special form of chromosomes found in the growing oocytes of animals except mammals. Polytene chromosomes were discovered by Balbiani, which are formed due to polytene and were observed in salivary glands of dipteran insects, because of which they are also called as salivary gland chromosomes. Now let us start off with the Lamprecht chromosomes. In the oocytic nuclei of those animals which have large yolky eggs, the prophase of the first meiotic division is extremely extended. During this phase, the oocytes grow and synthesize nutrients for the future embryo. In them, chromosomes become greatly enlarged and assume unusual configuration. A large number of loops project out from the chromatic axis, giving a Lamprecht appearance. Hence, these chromosomes are called Lamprecht chromosomes. The Lamprecht chromosomes are bivalent, each consisting of two chromatids which persist during the prolonged diploidine phase of meiosis prophase 1. The Lamprecht chromosomes history is they were first discovered by Fleming in the year 1882 in amphibian oocyte. J. Ruckert in 1892 had studied in detail about the Lamprecht chromosomes in the oocytes of sharks. Then Lamprecht chromosomes are found in the oocytes of insects, sharks, amphibians, reptiles, and birds, which produce large and yolky eggs. The size of Lamprecht chromosomes are large enough to be seen under light microscope. These may be as long as 1000 mu or more and about 20 mu in width. In salamander oocytes, they may attain length of about 5900 mu. Now let us see the detailed structure of the Lamprecht chromosomes. When we observe the Lamprecht chromosomes, they are in the diploidine stage. They consist of two homologous chromosomes which are in contact only at certain points, the chiasmata. Each chromosome of the pair is formed of two chromatids which are partial and form the chromosomal axis or the main axis. The axis is differentiated into alternative regions of high density loops which are highly colored and arise on both sides of the chromosomal axis. The chromosomal axis, the chromomeres, and the loop axis are all formed of DNA. The chromomeres are found in pairs and one chromomere on each chromatid. These are about 0.25 to 2 mu microns in diameter and are spaced about 2 mu microns from center along the chromatid axis. These probably represent heterochromatic regions where axial filaments remain tightly coiled. You can observe this in the figure that has been focused on the screen. Now coming to the each single loop, if we observe the lateral loops, they arise from the chromomeres either in two or in multiples of two. These extend on either side of the chromosomal axis about 5 to 50 mu microns and about 30 to 50 angstroms in diameter. Each loop consists of an axial fiber formed of DNA. It is surrounded with a matrix composed of RNA and proteins. That is why this gives a fuzzy appearance of the lateral loops. The Lamprecht chromosomes, when observed under the electron microscope, which was studied by Miller and Beatty in the year 1969 on these chromosomes of salamander oocytes, have shown the presence of dense granules on the loop axis of DNA. These dense granules represent large molecules of enzyme RNA polymerase. On getting attached to DNA, these initiate RNA synthesis. Arising from these RNA polymer molecules are seen fine fibers of RNA which increase in length. Each loop is considered to be one long operon consisting of a series of identical copies of the same structural genes, cistrons, which are separated by spatial DNA. Each gene locus probably produces a very long RNA molecule. This interacts with protein to form ribonucleoprotein. So this is all about the structure of the Lamprecht chromosomes found in the oocytes. Okay, now let us see how do the Polytene chromosomes look like. The Polytene chromosomes are of interphase chromosomes that are large and visible with the naked eye. These giant chromosomes are found in the cells of the salivary glands of Drosophila and Chironomus. In the cells of fat bodies of larval stages of certain Diptera, they are first observed in the salivary glands of Chironomus and hence are called salivary gland chromosomes. The Polytene chromosomes, as now I will be showing you the picture of the chromosomes and explaining the structure. Please do concentrate on the figure focused on the screen. A Polytene chromosome presents a distinct pattern of transverse banding. It consists of dark colored bands alternating with the light colored interbands. The bands are fuelgen positive, rich in DNA and therefore darkly stained. These are regarded to be chromomeres where DNA fiber is supercoiled. Adjacent chromatid fibers are aligned by their chromomeres forming dark bands. The bands are specific for a particular chromosome and banding pattern is almost similar in both the homologous chromosomes. While studying the relationship between the genes and the chromosomes, it was observed in 1981 that the structure of giant chromosomes is modified in certain regions due to enlargement and extension of the bands into lateral loops. Such enlargement or puffs are known as Balbiani rings or chromosomal puffs. The puffs form large rings around the chromomeres and are presumed to be formed by unwinding or uncoiling of chromonemata, which project out into a series of loops. These loops increase in size or diameter of the chromosome and this provides a fuzzy appearance like how we've seen in case of Lamprecht chromosomes. Here also the Polytene chromosomes also have this fuzzy appearance. If you see in detail the structure of the dark bands and the loops in puff very clearly, the formation of puffs is controlled by specific genes and takes place at definite time. These are concerned with the synthesis of RNA because besides DNA and protein, these contain large amounts of RNA. The puffs are chiefly associated with the metabolic activities of the chromosome because of high concentration of RNA. The process of puffing involves several a process such as the accumulation of acidic proteins, despiralization of DNA, synthesis of mRNA, and storage of newly synthesized mRNA in the chromosome. The puffs have been observed not only in giant chromosomes of salivary glands of Drosophila, but also on different chromosomes in different tissues of the dipterans. So, with this, we come to the conclusion of the topic giant chromosomes, in which we have seen how does the lamp brush chromosomes look like and how the polytene chromosomes look like. Both are the giant chromosomes, are of very important topic, often asked in almost all the exams. Thank you for watching my video, and please do like, share and subscribe. Hit the bell icon so that you get further notifications.